Good morning, everyone. In this video lecture, we'll learn about microencapsulation under the course of pharmaceutical technology. So let's start. Microencapsulation is a process by which very tiny droplets or particles of liquid or solid material are surrounded or coated with a continuous film of polymeric material. Microencapsulation may be defined as the process of surrounding or enveloping one substance within another substance on a very small scale, yielding capsules ranging from less than 1 micron to several hundred micron in size. So, in microencapsulation, one particle will be enclosed within a polymer, will be encapsulated within a polymer, and overall size would be 1 micron to 100 microns. Okay, so this would be micron in size. It is mean of applying thin coating to small particles of solid or droplet of liquid and dispersion. Microencapsulation is a process by which solids, liquids, or even gases may be enclosed in microscopic particle by formation of thin coating of wall material around the substance. So this is the main core material which contains our API, and this is the shell, okay, which is the sustained release polymer. So particle size could be from 50 to 5000 micron. So two phases, one is core material and another is coating material. The product obtained by this process is called microparticles, microcapsules, microsphere, coated granules or pellets. Particles having diameter between 3 to 800 micron are known as microparticles or microcapsule or microsphere, but larger than 1 millimeter are known as macroparticles. Okay. Here we can see there may have multiple layers. Layer 1, encapsulation layer 2, the core material. Okay, a well-designed controlled drug delivery system has been shown here. It can overcome some of the problems of conventional therapy. Then it can also enhance the therapeutic efficacy of a given drug. So our main purpose is to obtain maximum therapeutic efficacy. Drug is to be delivered to the target tissue in the optimal amount and in the right period of time. Thereby causing little toxicity and minimal side effect, because the drug would be delivered in a sustained manner. Okay, hence little toxicity would be shown. One such approach is using microsphere as carrier for drugs. Microsphere are characteristically free-flowing powder consisting of proteins or synthetic polymers, biodegradable in nature. Particle size is less than 200 micron. So fundamental consideration or formulation consideration for microencapsulation. Generally, microparticles consist of two components. One is core material, another is coat or wall or shell material. So core material, solid core can be a mixture of active constituents, stabilizer, diluent, excipients, and release rate retardants or accelerators. Coat or shell material, compatible, non-reactive with core material, provide desired coating properties like strength, flexibility, impermeability, optical properties, non-hygroscopicity, tasteless, and stable. So our core material contains solid or liquid, drug or active constituent, additive like diluent and stabilizer, whereas coating material contains different polymers, waxes, resins, proteins, polysaccharides. However, there may have some vehicles like aqueous and non-aqueous. So these are the three basic components for formulating microencapsulation. One is core material, that is our main API. Another is coating material that will ultimately encapsulate our product, encapsulate our API, because and the overall processing needs a vehicle. Okay, we will see in upcoming slides. So what could be our coating materials? I have put some coating materials um, which are as like as those materials needed for tablet coating. However, there are a basic role of polymer. Like polymers are substances of high molecular weight made up of repeating monomer unit. Polymer molecules may be linear or branched, and separate linear or branched chain may be joined by cross links. Polymer are used widely in pharmaceutical system as adjuvant coating material and a component of controlled and site-specific drug delivery system. So list of coating material. So here are water-soluble resins. Okay, PBP, CMC, methyl cellulose, arabinogalactone, polyvinyl acetate, polyacrylic acid. There are some water-soluble resins like ethyl cellulose, polyethylene, polymethylcrylate, silicone, cellulose nitrate. There are some wax and lipids like paraffin, beeswax, carnauba wax, stearic acid, stearyl alcohol. There are some enteric resin like shellegg, jane, cellulose acetate phthalate. So what are the basic reasons for microencapsulation? These are the some significance or rationale for delivering drug through microencapsulation: to protect reactive substance from the environment, to convert liquid active component into the dry solid system, okay, to separate incompatible component for functional reason. For example, if you have some incompatible substance, okay, then you can coat this with polymer to protect the immediate environment of the microcapsule from active component. Isolation of core from its surroundings, as in isolating vitamins from the deteriorating effect of oxygen. Okay, for example, sometimes vitamins are provided in that manner because vitamins are very susceptible to oxygen. Then retarding evaporation of the volatile core, improving the handling property of sticky material, or safe handling of toxic material, because your API can be toxic, and in that case, to coat it with non-toxic polymer is very important. To get targeted release of the drug, for example, colon-specific drug delivery system, to control the release of the API for delayed release or long-acting release. The problem may be as simple as masking the taste or odor of the core, to increase the bioavailability to produce a target drug delivery, to protect the GIT from irritant effect of the drug, extension of duration of activity for an equal level of active ingredient. However, there are some pharmaceutical applications for microencapsulation, like formulate various sustain and control doses from, formulate enteric coated doses from, reduce the chance of gastric irritation, taste of bitter drug can be masked, liquid gases can be changed into solid particle through microencapsulation, and it can be employed to aid in the addition of oily medicine to tableted doses from, to overcome the problem of tachygranulation, and indirect compression can be used to decrease the volatility. Okay, it can provide environmental protection to the encapsulated active agent. It can reduce the hygroscopic characteristics of many core material. Okay, so these are the common applications of microencapsulation. The separation of incompatible substance can be achieved. Okay, it can be used to lessen the potential dangerous toxic substance handling. Okay, so let's discuss the classification of microencapsulation technique. So these are the methods that we should learn about. There are some physicochemical or physico-mechanical method, some physicochemical method, okay, some chemical method. In physical or physico-mechanical method, uh, no chemical reaction takes place, just it's the processing or physical method, like air suspension, centrifugal extrusion, paint coating, spray drying, okay, vibration and nozzle. We'll discuss this later. However, there are some physico-chemical method like inotropic gelation, coacervation. We'll also discuss this. And in chemical method, where some chemical reactions takes place, it includes solvent evaporation, polymerization, interfacial polymer, in situ polymerization, then matrix polymer. So these are the common classification of microencapsulation technique. So first of all, air suspension. So this process is as like as tablet coating method by Wattstad process. Just the main difference is that in tablet coating or in granulation, we usually perform this for granules, but here we usually provide the core material. This is the basic difference. Otherwise, the overall process is similar. Okay, so here you can see air distributed plate from where air would be provided, because the uh, core would be suspended here. This is a vacuum place. Okay, then hydraulic or pneumatic nozzle. This nozzle will provide some spray. Okay, this nozzle will provide some you need to provide polymer. Then coating spray, yeah, this will provide your core material. Then airflow, the coating partition. Here the partition takes place because the granules or the core would ascend in that way, but whenever they would come here, due to the airflow pattern, they will come in this direction once again. So this will be continuing one by one, and your overall uh, core would be coated. Then control particle flow, you can see the direction of the flow. Okay, so what are actually happening, you are providing the solution, the polymer and the core throughout this way, then they are getting suspended in the air, and that is why this process is called air suspension. And after that they will again come down into the surface. Okay, and this process will be continuing one by one until or unless your prepared micro encapsulation is formed. So solid particulate core material are dispersed in a supporting air stream. The coating material is spread on the air suspended particle. Within the coating chamber, particles are suspended on an upward moving air stream. The design of the chamber and its operating parameters affect a recirculating flow of the particles through the coating zone portion of the chamber, where a coating material, usually polymer solution, is spread applied to the moving particles. During each pass through the coating zone, the core material receives an increment of coating material. The cyclic process is repeated perhaps several hundred times during the processing, depending on the purpose of the microencapsulation. The coating thickness desired until the core material particles are thoroughly encapsulated. The supporting air stream also serves to dry the product while it is being encapsulated. So schematic of a fluid bed coater, it can be of top spray or bottom spray or tangential spray. In previous figure we have seen the bottom spray, okay, which is also called word stirred process, but spray can be done from upwards direction, okay, or it can also be done from tangential direction. Drying dates are directly related to the volume parameters of the supporting end stream. Then spray drying and spray congealing, so this is another method for microencapsulation. So we have also learned this in our granulation chapter, because spray drying is another process of tablet coating, okay, granulation coating. So this process is as like as spray drying of microencapsulation. And another one is spray congealing. Actually both of them are similar, just main difference is their parameter. We'll see this in upcoming slides. So the coating solidification is affected by rapid evaporating of solvents in which coating material is dissolved. Microencapsulation by spray drying is a low-cost commercial process which is mostly used for the encapsulation of fragrances, oils, and flavors. Steps: core particles are dispersed in a polymer solution and spread into the hot chamber. The shell material solidifies into the core particles as the solvent evaporates. The microencapsulated obtained are of polynuclear or matrix type. So usually spray drying is nothing but the spray drying of the tablet coating. This process is similar as previous one. Okay. So here we can see the overall steps and procedure for spray drying and spray congealing. First of all, air is intake, then heater is applied, issues the nozzle, and from this nozzle, your uh, polymer and coating material are sprayed in that location. So due to the heated air, they would get dry. Okay, so here is a temperature sensor which will sense the temperature and the cyclone formed. Okay, then here is the collecting vessel. This dried microencapsule would be collected here. So one question may arise then, where does that heated air will go? Actually, the heated air will go in that way by the aspirator. Air is intaking and air is getting out. So there is no phenomenon regarding the air. Air has just performed its function regarding the drying. The function of the heated air is drying, nothing else. Okay. So this is the overall process. The main difference is that for spray drying, aqua solution or hot air is used. But think, just if we would use cold air, okay, and we would use here hot melt solution. It's not actually solution; it's some sticky material, hot melt, instead of aqua solution. Then this process would be spray congealing. So the overall process here, which has been shown, is spray drying, but in case of heater, if you remove the heater and provide and this as uh, hot melt other than aqueous solution, then this process would be spray congealing, and other figures and procedures are same, nothing is different. Okay. So spray congealing, the coating solidification is affected by thermally congealing a molten coating material. The removal of solvent is done by evaporating technique. This technique can be accomplished with spray drying equipment, which is when the protective coating is applied as a melt. So actually this process is as like as spray drying. First of all, the core material is dispersed in coating material melt. This is very important. You have to provide the melt. Coating solidification and microencapsulation is accomplished by spraying the hot mixture into a cool air stream. Microencapsulation of vitamins with digestible waxes for taste masking. So there are some similarities between spray drying and spray congealing. So both processes involve dispersing the core material in a liquefied coating substance or spraying into the introducing the coating material onto the core material. Both processes involve solidification of coating material. The principal difference between the two methods is the means by which coating solidification is accomplished. Both processes have advantages such as low bulk density product, porous nature, capsules, and free-flowing particles. Then solvent evaporation. In the case in which the core material is dispersed in the polymer solution, polymer shrinks around the core. In the case in which the core material is dissolved in coating polymer solution, a matrix type microencapsule is formed. The core material may be either water or water soluble or water insoluble material. A variety of film forming polymers can be used as coating. For example, evolution of sucrose esters as alternative surfactant in microencapsulation of protein by the solvent evaporation method. So, solvent evaporation process, core material, it is dissolved or dispersed in coating polymer solution. Then with agitation, liquid manufacturing vehicle phase is performed, and upon heating, evaporation of polymer solvent and microencapsulation. Actually, you will apply some polymer, some tablet core, and some solvents, and this solvent would be evaporated, nothing else. Okay, so you can see the overall process here. First of all, API, polymer, and organic solvents has been given here. Then you will stir them for agitation or mixing. After that, solvent would be evaporated. You can see here the solvent has been evaporated, and when all the solvent would be evaporated, the polymer will enclose the API or the core. Okay, and your ultimate result would be like that. So these are the microencapsules. So here step-by-step procedure has been shown. First of all, formation of solution or dispersion of the drug into organic polymer phase, then emulsification of the polymer phase into the aqueous phase containing a suitable stabilizer, thus forming a O by W emulsion, and finally removal of organic solvent from dispersed phase by extraction or evaporation leading to polymer precipitation and formation of the microsphere. So these are the overall procedure for solvent evaporations. Next, our another procedure for another method for microencapsulation is polymerization. As the name suggests, polymerization. So several monomer will ultimately get attached with each other to form a polymer. Okay. So a relatively new microencapsulation method utilizes polymerization technique to form protective microencapsule coating in situ. The method involves the reaction of monomeric unit located at the surface interface existing between a core material substance and continuous phase in which the core material is dispersed. The core material supporting phase is usually a liquid or gas, and therefore polymerization reaction occur at liquid gas or solid liquid or solid gas interface. In the formation of polyamide, polymeric reaction occurring at liquid-liquid interface existing between aliphatic diamine and dicarboxylic acid halide. So types of polymeric reaction, it can be of three types like interfacial polymer, in situ polymerization, and matrix polymer. So interfacial polymerization actually two reactants. Interface means it's some kind of interface. Okay, interface exists between uh, two things. Okay, then in situ. In situ means actually in place. Okay, in the actually situated. In situ is related to situated. Actually, in the location, in the same location. It means okay. That means in the reaction vessel, this polymerization will happen. And another one is matrix polymer. That is the you know your API would be dispersed within matrix. Okay, so in interfacial polymerization, the two reactants in a polycondensation meets at the interface and react rapidly. Then in situ polymerization, in a few microencapsulation process, the direct polymerization of single monomer is carried out on the particle surface. That is, cellulose fibers are encapsulated in polyethylene while immersed in dry toluene. Usual deposition rate are about 0.5 micron per minute. Coating thickness ranges from 0.2 to 75 micron. And last one is matrix polymer. In a number of processes, the core material is embedded in a polymeric matrix during formation of the particle. Prepared microencapsules containing the protein solution by incorporating in the aqueous diamine phase. National Lead Corporation utilizing polymerization technique. So overall process of polymerization has been shown. That is pre-polymer, that is surfactant. Okay, this is the oil. So water phase is added. Then upon the mixing, this type of emulsion will be formed. Then polymerization will be happened. You can see several monomers. Monomers are dot dot sign. So upon the help of surfactant, they are getting added with water. You can see here. Okay, they are separated now. The water droplet and the green polymer is separated, and the surfactant will attach them. Okay, so both of them has been attached, and the ultimate microencapsule would be like that. That is encapsulated. There is no encapsulation, but here is encapsulation. Then coacervation phase separation. It involves three steps like a formation of three immiscible phase, deposition of coating, rigidization of coating. Okay, so polymer homogeneous solution, then phase separation takes place. The coacervate droplet is here, and after some time they would coat it. Okay, they would coat it with the droplets, and ultimate result would be polymeric membrane. Actually, three steps are happening, as the name suggests. Coacervation means something like you know your core material phase, then coating material phase, and liquid polymer manufacturing phase would be coacervate with each other. And you can see the coacervate droplets. So formation of three miscible phase, and that is why it is called in coacervation, because the main criteria for coacervation is that three of them should be immiscible. If one or two of them is miscible with one another, then it would not be coacervate. Okay, so formation of three miscible phase: a core material phase, a coating material phase, and a liquid manufacturing phase. Initially, the core material is dispersed in a homogeneous solution of coating material in which solvents for solubilizing the polymer in the liquid manufacturing vehicle. The coating material is separated from the liquid manufacturing vehicle to form coacervates by application of any of the following principle. For example, it can be done by changing the temperature, addition of the salt, addition of non-solvent, addition of incompatible polymer to polymer solution, then inducing polymer-polymer interaction. So because of the application of the above principles, the solubility of the polymer in the liquid manufacturing vehicle decreases, and the polymer starts separating in the form of small liquid droplets called coacervates, thus resulting in the formation of three phases: a liquid manufacturing phase, a core material phase, and coating material phase. Then deposition of coating. The deposition of coating material takes place when the coacervates droplets starts arranging themselves and get absorbed over the surface of core material, resulting in the formation of core around the core particle. Rigidization of coating, and by this rigidization actually your coat would be rigid. Okay, the coating is made rigid by temperature rigidization and cross-linking, rigidization, then dissolution, evaporation of the solvent. So this is the overall procedure for coacervation. However, there is another method like pen coating. Actually, this pen coating is as like as tablet coating. It is one of our conventional methods for tablet coating. This pen coating, we keep the core in a conventional pen, then we spray. Okay, upon the spraying and the rotation of the pen, Actually, the coating material will encapsulate the droplet. Okay, so I'm just going through this. So this is the pen coating. Just go through this. You can easily learn this. However, I have described uh, full pen coating mechanism in tablet coating. Please, if you want to have a broader description, please go through the slides. So pen coating principle, pen speed is a factor, degree of atomization is a factor, spray pattern is a factor, then coating material is a factor. Okay, there are number of factors. Okay. Temperature is a factor, spray pattern is a factor. So these are the factors of pen coating, which has been discussed broadly in tablet coating video. Okay, just go through this. However, there are some factors which will impact encapsulation efficiency, because sometimes your encapsulation may be very robust and sometimes it is not so much good. Okay, actually this will depend on some factors. Here you are using some polymers, you are using solvents, you are using some you are applying temperature, you are evaporating this, you are drying this, then you are mixing this, your agitation, all of them are factor. You know, all of them should be optimized. Okay, your pH is another factor. A lot of factors are behind this encapsulation efficiency, and if you can optimize all of the factors, then your encapsulation would be very efficient. Okay, so to have better understanding, I have provided the positive, the proportional one as the blue one and the inversely proportional one as the red one. Okay, first of all, solubility of the polymer in organic solvent, it is inversely proportional. Why? Because just think, if your polymer is very much soluble in organic solvent, then what will happen? It will, is your polymer would not react with the core, with the core material, if it is very soluble with the organic solvent. So please make sure that your polymer is less soluble in organic solvent, otherwise it would not get encapsulated within the core. So here it has been said. Then solubility of organic solvent with water. Obviously, water and organic solvent, these are vehicle, and they should be soluble with one another. Otherwise, evaporation will not take place, because high solubility on water means faster solvent removal and faster evaporation and high encapsulation efficiency. Okay. Then concentration of the polymer. You know, the better polymer would you have, the better would be your uh, microencapsulation. So higher polymer concentration, increased viscosity, faster solidification, high efficiency. Then dp by cp ratio, that is dispersed phase by continuous phase ratio. Okay, actually it means higher volume of the continuous phase, the faster solvent extraction, higher encapsulation efficiency. You know, your continuous phase should be higher. For example, your microencapsulating, and in your microencapsulation process you have kept your continuous phase very low, you have kept your solvent very low, then what would happen? Your continuous phase would get decreased. Okay, actually this should not be too low or should not be too high. Just keep it in your mind. Okay, so the dispersed phase and the continuous phase ratio should be uh, lower one. Okay, because whenever it would be low, that means your continuous phase should be very high, and if your continuous phase is high, that means your overall microencapsulation will be very good, because you cannot perform your uh, your uh, microencapsulation within a very low solvent. Okay, so that's why dp by cp ratio should be low, as low as possible, because this is low means your continuous phase is high, and more high the higher your continuous phase, the higher encapsulation efficiency you would have. However, then solvent removal rate, obviously your solvent removal rate should be higher, because otherwise you have to wait for days, months, years for just microencapsulation. So solvent removal rate should be higher. Then interaction between the drug and polymer, obviously your interaction should be very good. Okay, strong affinity, there should have some strong affinity of the polymer with the core material, otherwise your encapsulation will not be very good. Then molecular weight of the polymer, so low molecular weight polymer generally give higher encapsulation efficiency. This is actually related to the grade of the polymer also, because higher molecular weight may cause inhomogeneous drug distribution and lower efficiency. because for example, this is your code, tablet code, okay, and you will apply some polymer, but its density is very high. Whenever its density very high, then what will happen? They will get spreaded in that manner, okay, they will not come in that direction, they will not disperse within the core homogeneously, because this is very sticky, this is very dense, this is with high molecular weight. Your encapsules are, your polymer is very high molecular weight, it would not try to get spread. But just think another case, if you have a very uh, low molecular weight polymer, then what will happen? For example, this is a low molecular weight polymer, like watery polymer, then it will easily get spread within your core. This is the basic difference between high molecular weight and low molecular weight polymer. And last one, solubility of the drug in continuous phase. Obviously, what do you want? Your drug is very much in cooperation with continuous phase. Your drug is not interacting with polymer. Then obviously this will negatively impact your microencapsulation efficiency. So yes, solubility of the drug in continuous phase should be lower. Otherwise, your drug would not be interacting with your polymer. So high drug solubility in the continuous phase means greater drug loss and lower encapsulation efficiency. So this is the overall factors regarding encapsulation efficiency. However, we have prepared our microencapsule by various techniques, methods, and it's time for evaluation. So how we would evaluate our microencapsule? Actually, this evaluation is as like as tablet evaluation or capsule evaluation, because microencapsule is also a solid dosage form. Okay, and the evaluation will be like content uniformity with variation, some dissolution test, disintegration test. Okay. So the evaluation of microcapsule. Moreover, you can also evaluate this by scanning electron microscopy, X-ray crystallography, DSC, a lot of things. Use FTIR, a lot of process. Okay. First of all, percentage yield, then scanning electron microscopy. I'm just going through this. For details, please read on this. You can easily understand what we'll done. Then particle size analysis. Obviously, particle size analysis is also important, because uh, in which micrometer it is, we should analyze this. If it is in millimeter, then obviously it would not be microparticle, so very carefully analyze this. Then encapsulation efficiency is determined by actual drug content divided by theoretical drug content into 100%. Then in vitro drug release, I have discussed all of this in the tablet evaluation. Please go through this. You know, you record ESP type 2 dissolution test apparatus, pH should be adjusted, temperature should be adjusted, pedal speed should be 100 rpm. Okay, then please go through your uh, dissolution test. Then uh, all studies are conducted in triplicate. Okay, then take the sample and investigate this under UV spectrometer. Okay, so these are the basic steps. So estimation of the drug content, that means the potency or the assay. It's just you can do that by UV spectroscopic method. Just prepare a sample and just go through this in 254 nanometer in UV spectrophotometer. So what are the advantages of microencapsulation? Actually, I have already discussed this in the reasons for microencapsulation in the very beginning of the video, but just go through this. Formulation of the sustained release dosage form, providing protection, conversion of liquid and gas into solids, masking the beta test, reduction of the hygroscopic nature, reduction of the vaporization and loss of volatile drug, improved stability and longer shelf life, targeted drug delivery, reduced side effect and toxicity, improved patient compliance. However, there are some disadvantages also, because it is one of the very costly method. It is very costly than tablet or capsule manufacturing, okay, or any other doses from solid doses from manufacturing. This is very costly, and you know, the process of manufacturing is not so much easy. These are complex manufacturing process. Okay, so expensive and complex technique. Coating may not be uniform, affecting the drug release. Possible cross reaction or incompatibility between core and shell material. Requires high skill, specialized equipment, and technical expertise. Scale up from laboratory to industrial production is often difficult. Residual organic solvent may remain in the final production. Sometimes low drug loading capacity. Burst release or incomplete release can occur in the some formulations. Regulatory hurdles due to complex manufacturing process. So these are the applications, rationales, significance, importance, disadvantage, advantage, and some methods of microencapsulation. If you still have any confusion, please inform me in the comment section. Thank you.